So yesterday we had discussed on the way the prokaryotic cell, okay, such as a bacterial cell, we have seen that how the E. coli cell, which is a bacteria, they regulate the expression of gene by using the units of DNA we call operons. So they use these operons for regulating the gene expression. So generally speaking, an operon is a segment of DNA we have studied yesterday, right? It is nothing but it is a segment of DNA molecule that not only contains the specific structural genes that are needed to produce some types of proteins or enzymes, but we have seen yesterday that it also contains other regions, other segments, such as the operator, right? The operator as well as the promoter section. Besides the structural gene, where the cell used to use this segment of DNA to transcribe into mRNA molecule. So we have studied that a DNA molecule, it does not only contain the structural genes required to be transcribed into mRNA molecules and finally produce proteins, but it also contains all other segments such as the promoter as well as the operator. So all this we have studied yesterday, and now we will focus on the type of operon, okay, that is used by a bacterial cell in anabolic processes. Okay, that is used by a bacterial cell in anabolic processes. Now, what are anabolic processes? They are simply a process in which, okay, simply a process which synthesize some types of molecules, okay, some types of molecules needed by the cell. And the type of operon that we are going to focus today is nothing but a repressible operon. And yesterday, if you remember, we had studied on an inducible type of operon. Okay, example here we have studied the lac operon. And we have also seen yesterday that the lac operon, it remains, okay, in a switched off condition. In a natural condition, it remains off. Only when a certain inducer is being added to the cell, to the bacterial cell, okay, from outside, then only this operon will switched on. But today we are going to study on another type of repressible operon, okay, which is required, that is used by a bacterial cell in anabolic process, meaning in those process which synthesize some types of molecules needed by the cell. So like I said that here the repressible operon, okay, is an operon which is used by a bacterial cell, okay, which is used by a bacterial cell in anabolic processes. And anabolic processes are those processes which synthesize the required which synthesize certain types of molecules required by the cell. So here an example of a repressible operon that we are going to study is a tryptophan operon. Now you have studied about the amino acids, right, in your fifth semester, so now you know why, you will see why this operon is given the name tryptophan. Okay, tryptophan operon. Just as yesterday we have studied that lac operon because the inducer here is lactose. That's why the name of the operon has been given as lac operon. So same thing here, Now, a specific example of a repressible operon used by a bacterial cell is a tryptophan operon. And a repressible operon is the most common type. Okay, it is the most common type of operon used by bacterial cells in anabolic processes. And also remember one thing here, the lac operon, it usually remains off, right? It will be on only if certain enzymes are required to be synthesized. But this is exactly the opposite of lac operon. The tryptophan operon is usually on. Okay, it is usually on in natural condition. But sometimes under certain circumstances it can be turned off. So this tryptophan operon is usually on, but it can also be turned off. We will study about this, how and when the tryptophan operon is turned off. Okay, so a tryptophan operon is an operon used by bacterial cells to basically regulate the synthesis of tryptophan. Okay, the synthesis of tryptophan amino acid. So that is why the operon here, it has been given the name tryptophan operon, because this operon is used by bacterial cells to basically regulate the synthesis of a tryptophan amino acid. Now, if we take a look at the different components of a tryptophan operon, okay, a tryptophan operon, it contains, okay, of the regulatory gene, okay, or the repressor gene, followed by the promoter gene, and this segment of gene is the operator, okay, which will operate the transcription process, and followed by the structural genes, which will be transcribed into mRNA molecule. Now, the structural genes which are usually required, okay, for producing those enzymes or proteins, okay, which are used in the synthesis of a tryptophan amino acid, okay, 5 different types of genes, okay, there are 5 different types of genes required to produce 5 different types of enzymes, and these are the enzymes which involve in the biosynthesis of a tryptophan molecule. So here, to produce these 5 enzymes, there are 5 different structural genes which will be transcribed, which will code for the enzyme, the 5 different enzymes required in the biosynthesis of a tryptophan molecule. So just remember the structure. Unlike the lac operon we have seen yesterday, it has the inhibitor, the promoter, the operator, and 3 structural genes required to produce those 3 enzymes to metabolize lactose, right? Yesterday you have studied this one. So here in this case, just remember, in a tryptophan operon, we usually have 5 different types of structural genes which will code for these enzymes required for the biosynthesis of the tryptophan amino acid. So girls, let us take a look at the first condition, okay, where this operon, the tryptophan operon, is turned on. Now, what happens when the operon is turned on? This condition it occurs when the tryptophan in the cell, okay, it is low. The concentration of tryptophan amino acid in the cell is low. Then the repressor protein, okay, is inactive. Now we can see here there is a repressor gene, okay, a repressor gene. This is a promoter, and this one is the operator. So what happened actually in a natural condition in the cell, the tryptophan amino acid is usually low, the concentration. So in this case, when the repressor gene, it will transcribe, it will produce the repressor mRNA molecule. And this repressor mRNA molecule will be translated to form repressor protein. And remember in this case that the repressor protein is inactive. You should always remember this, that it usually inactive. That means there must be something, okay, to make this repressor protein active so that it can bind to the operator. Just like in the lac operon, we can see clearly that the repressor protein, which is produced from the repressor mRNA, from the repressor gene, the repressor protein in lac operon, it is active and it can bind the operator and block the transcription process. But in tryptophan operon, when the tryptophan concentration is low, this repressor protein produced is inactive and it cannot bind to the operator. So now the operator is free. If it's free, then suppose this is the RNA polymerase enzyme, okay, it will bind to the promoter and it will slide down along the gene, synthesizing, transcribing the mRNA molecule and synthesizing these enzymes. Okay, that means here the transcription process will take place and finally translation, which will form the required enzymes, and these enzymes, they will synthesize the amino acid tryptophan. Okay, so the process here, we say the operon here is said to be on because the RNA polymerase, it can bind to the promoter and there is nothing which is blocked in this operator molecule, so it will slide down here along the gene until it reaches the end of the gene, okay, this RNA polymerase, and it will synthesize these. Finally, the tryptophan amino acid will be synthesized. So this is the first case which occur in the cell, the natural condition when the tryptophan concentration is usually low. But what happened in the second condition when the tryptophan concentration is very high in the cell? So just in case the tryptophan concentration is too high in the cell, what that excess tryptophan will actually go where? It will actually bind to the repressor protein, to the allosteric site of a repressor protein. Okay, what happened when it binds to the allosteric site? As you can see here, this is a tryptophan amino acid. Because it is low in concentration, so it is not able to bind here in the repressor protein. Whereas when it is in excess or the concentration of tryptophan is too high, those molecules of tryptophan, they can actually bind to the repressor protein and thus it acts as a co-repressor. Now when a tryptophan and a repressor protein bind, this repressor protein now becomes active. Here it is inactive and now it becomes active after the binding of tryptophan, and finally it can go and bind to the operator gene. Now we can see here the operator gene is free, whereas here in this case the operator gene has been blocked. So the RNA polymerase which bind to the operator, it cannot go beyond the gene, it cannot go further, and therefore no mRNA molecule is formed. So this is said to be the operon now is said to be turns off because no mRNA is produced and no enzymes is formed, and now no more tryptophan amino acid is produced. Okay, because the tryptophan is already in excess, so it blocks the process, it turns off the operon. So girls, I hope it's clear enough that when tryptophan is low, you can see that the the structural genes will be transcribed into mRNA and finally translated into proteins, which involve in the biosynthesis of tryptophan amino acid. And in case there is so much of tryptophan concentration in the cell, when it is too high, then these tryptophan, they will uh, bind to the repressor protein, making it active, and thus this active repressor protein now will block the way of the RNA polymerase enzyme by binding to the operator site. So it is as simple as that. Okay, when the tryptophan concentration is low, the operon is on, whereas in case the tryptophan concentration is high, the operon turns off. So here one more thing I want to um, explain about another important, very important process is tryptophan attenuation. Okay, the attenuation process. What is this? This is very important, girls, for you to understand. Okay, so here what happened actually, sometimes even after the repressor protein it binds to the operator, sometimes the transcription process can still occur. Okay, that means the RNA molecule can still slide down the gene through the gene and continue with the transcription process, and the transcription will occur and the tryptophan will continue to be synthesized. So this is dangerous for the cell, for the bacterial cell, because why? In this case we know that the tryptophan concentration is already very high. So if the RNA polymerase continue to transcribe the genes, then it will be dangerous for the cell. So in this case, what happened, even though the repressor protein may be due to some problem, okay, maybe the repressor protein, it gets detached from the operator and the RNA polymerase can still continue with the transcription process, whereby the tryptophan concentration is already high. So in this case, the bacterial cell, it will there is another process by which a bacterial cell will regulate this tryptophan biosynthesis, okay, through the process termed as attenuation. So we will take a look at this process. What is this tryptophan attenuation? So girl, let us take a look at this tryptophan operon attenuation process. Now in this tryptophan operon attenuation process, okay, that means attenuation is the process of terminating the synthesis of those structural genes required for tryptophan. Okay, that means it is the process of tryptophan synthesis halting. So here now let us try to understand this process. We know that here in the first place we have the repressor gene, right? The repressor gene, and we have the promoter, the operator, and these are the 5 genes, okay? E, Trp E, Trp D, Trp C, Trp V, and Trp A. So, and other genes, okay? We know that of course the DNA molecule is long, so it will continue here, but these we will concentrate on these 5 genes which are required for the biosynthesis of tryptophan. So we know that in a tryptophan operon we have these segments of genes. Now I did not mention to you about this portion before, okay, because I want you to concentrate on the how the tryptophan operon is turning on and off. But now, like I said, just in case this process it fails, okay, that means even if the tryptophan concentration is very high in a cell, sometimes the RNA polymerase which bind to the promoter, it can still continue with the synthesis of these enzymes, okay, even though the repressor protein from which we get from this gene, the repressor protein, after it already binds to the operator, still the transcription can still continue. So in that case, this attenuation process, it comes into picture. The bacterial cell, the E. coli cell, it will use this process, attenuation process. Now, here for understanding this attenuation process, you must understand about this segment here. This segment, actually, it is a leader segment. Okay, it is referred to as a leader segment, and it's present just before the genes, just before these 5 genes. Okay, the structural genes required. That means in a structural gene, we have these 5 segments of genes, okay, which will form those 5 different enzymes, okay. But just before this, we have a leader sequence. That means the RNA polymerase, it will have to go first through this leader sequence in order to transcribe these genes. Now, what happened even after the repressor, it has bind here the repressor protein, but still the RNA polymerase, it will continue to transcribe. What happened when it binds to the promoter, it will slide along the gene. We know that, right? It will slide along the gene and it will reach this leader sequence. So here, let us just enlarge the picture of this leader sequence, okay? Suppose this is the leader sequence here. I will just enlarge this portion for you to understand. Now, if I enlarge this portion, actually only the small portion here I have enlarged. The leader sequence, just remember, it has 4 segments. That means even in the leader segment here, we have 4 segments. The first segment, second, third, and fourth. Now, these 4 segments which are present in the leader sequence, okay, just remember, they are complementary to each other. They have the palindromic sequences. If you remember, palindromic sequences, they can base pair with each other because they are complementary, right? Suppose here if I have UGG in first, okay, then in second I will have a segment as ACG. Then these are complementary, right? So they will form base pairs. They will base pair with each other. So that means these segments, they are complementary with each other. One is, it has segments which are complementary with 2. It has a complementary sequence with 3 with 4, and like that. Okay, all these 4 segments are made in such a way in a leader segment so that they can be like this. Now, what happened actually? We know that in a bacteria, we know that both the transcription and translation, okay, they takes place alternately. They takes place at the same time. That means as soon as the mRNA is being synthesized, only even a short segment of mRNA is being synthesized from this DNA, is being transcribed from this DNA. At that time, the ribosomes, remember, the ribosomes will come and bind to this short segment of RNA, and they will start to translate into proteins. Now, guys, girls, remember that this mRNA is still being transcribed from the DNA. Suppose the RNA polymerase is still going through from here, from this segment. It transcribed this mRNA molecule, and translation also, it takes place at the same time. So what happened when one base pair with 2, okay, these segments, suppose they form a loop. Now some of the sequences here, they form base pairs. Suppose 1 and 2, this actually will act as a pause signal. Okay, they will give a pause signal to the transcription process. but this is nothing to do with attenuation. Now what happened when 2 and 3 they form a dimer? They will form an anti-termination signal. Okay, and when this is 2 and 3, and when 3 and 4 they form a dimer, okay, they will produce a termination signal, termination of translation. So we know that in a leader sequence, of course, this is a double-stranded DNA molecule. So this base pairing, it will not cause between these segments, it will not be present in the DNA molecule. It will not occur, sorry, in the DNA molecule, because it is already a double-stranded. Of course, this will be when the mRNA molecule is being synthesized. Okay, that means suppose the RNA polymerase, it has reached this region, and the mRNA is produced. So in the mRNA also, okay, we have this. Suppose this is the portion of mRNA which is formed from the leader sequence. So we know that the leader, it has these 4 segments. So in this mRNA, we have these segments 1, 2, 3, and 4. Right? So because it is single-stranded, then it can fold itself. So 1 and 2 can form base pair. Okay? So they will give a pause signal. 2 and 3 they will give an anti-termination signal, and 3 and 4, if they form a base pair, okay, if they form a dimer, if they base pair with each other, then they will be giving this termination signal, termination of translation. So what happened actually, as the transcription process still occur, and some of the part of the leader segments, suppose it has being synthesized, okay, this mRNA molecule, it has being synthesized, small portion. Now the ribosome will come and bind, right? Will come and bind first to this first region. Now girls, just remember, in this first segment, okay, of the leader sequence, we have UGG, UGG. And this UGG codon, remember, it is known as a codon. 3 nucleotides are referred to as codon. So this codon is actually it will actually code for tryptophan. So you must be thinking, when these enzymes are needed to be produced, so that they will take place in the biosynthesis of tryptophan. Now why the cell itself, this mRNA itself, it has these codons by which can be translated into tryptophan, which codes for tryptophan. Why? So you will understand this when we take a look at the whole process of attenuation. Now what happened here actually, suppose this RNA polymerase, okay, it has moved along this DNA molecule, and it reaches the leader sequence, this one I had shown here. Suppose it finished to transcribe this segment 1 and 2. When it finishes to transcribe segment 1 and 2, okay, suppose the RNA polymerase, it reaches this one, the leader sequence, and it finished to transcribe these 2 segments. Then these 2 segments, they will form a loop, right? When they will form a loop, they will give a pause signal to the RNA polymerase. At that time, the ribosome, okay, the ribosome will bind to this mRNA, okay, which has been transcribed, will come and bind here, okay, and will come and bind and will start the translation process. As we know that in bacteria, both transcription and translation, they takes place in the simultaneously. So when the ribosome, they come and join here. Now what happened when the ribosome join in the first segment, they will come across these 2 codons, and they know that these 2 codons, when the ribosome, they come and bind here, they will ask the tRNA molecule to bring the tryptophan, because these are the codon for the tryptophan amino acid. Then in that case, what happened? Suppose in the cell we have a very low concentration of tryptophan. Okay, we have a low concentration of tryptophan, so it will take time for the tRNA molecule. Remember, the tRNA molecule, they will bring the amino acid along with them. So since there is a low tryptophan concentration in the cell, then it will take time for the tRNA molecule to bring the tryptophan, okay, here in the ribosome machinery. So in that case, it will take time. This ribosome will halt, okay, it will stay here in the first sequence. Now the RNA, it starts synthesizing all these other sequences. Suppose 2 and 3 also, it finished to transcribe from this DNA. Now what happens, since the segment 1 is not free because the ribosome is binding to the sequence 1, now 2 and 3 will base pair with each other. Those complementary segments will base pair with each other. And what happens when they base pair with each other? They will give an anti-termination signal. An anti-termination signal. That means they will ask the translation process to continue, okay, because they are anti-termination. They won't terminate the translation process. They will give a signal for the translation to occur. Now when the, suppose in a that is the condition when the tryptophan is low in a cell, all right, because the ribosome will stay here in segment 1, then 2 and 3 will form a pair because this segment 1 is not free. So in this case, we get an anti-termination signal. That means the translation process can continue. Then these enzymes, these 5 enzymes, okay, can be produced and they can takes place in the biosynthesis of tryptophan. So because of the low tryptophan, that means the process, the operon itself is on. But girls, just imagine if there is a high concentration. When the concentration of tryptophan is high, then the cell itself, it does not need to produce more tryptophan. So in that case, it will try to off the operon. So how this process will take place? Okay, the attenuation process. Now, how it will takes place in this case is that now since the tryptophan content is very high, so when the ribosome, it binds to the first segment, okay, it comes across these codon, and now it will ask the tRNA molecule to bring the tryptophan. So because of the high concentration of tryptophan in the cell, it will not take time to bring the charged tRNA molecule, okay, this tRNA molecule with the tryptophan amino acid, it will bring it to this, then translation process can occur. Now in that case, this will move fast, okay, the ribosome, it will not halt in this segment 1, it will start to move at a very faster rate, and it will reach the segment 2 and the translation process will continue. So in that case, when it reaches the segment 2, now the segment 2 is not free, okay, but now the RNA molecule, the RNA polymerase, it finished to transcribe all the segments 3 and 4 also. So now since 2 is not free, so 3 will combine with 4, will form a dimer with segment 4. Now I had mentioned that when 3 and 4, they form a loop, okay, they form a dimer. Then in that case, as soon as they form this structure, the dimer, due to their sequence, okay, which are complementary, they will base pair and they will form this dimer. In this case, they will produce a termination signal. So when they produce a termination signal, that means they will give a signal to the cell to stop the translation process. Why? Because when 3 and 4 combine, remember, they will give a termination signal of translation. So translation will stop. So that is the case when there is a very high concentration of tryptophan. Then we know that the ribosome, it will not halt in this first segment, it will move quite fast. So when it reaches the second segment, then at that time the 3 and 4 can combine, since segment 2 is not free, and they will give a termination of translation signal. So the translation process will stop. In that case, when it stops, okay, actually the whole transcription process also will stop. The RNA polymerase, before it reaches this part, okay, before it transcribes the structural gene, the whole process will stop due to the signal from here. So this is all about the attenuation process, the tryptophan operon attenuation. So girls, I hope you understand, because here there are two mechanism. Okay, one is through this mechanism by which the tryptophan itself, when it is high, it will bind to the repressor molecule. But what happens when this process, when this mechanism fails, then the bacterial cell, the E. coli cell, they will apply this attenuation process.